First of all, I'd like to thank Katrina for organising this exciting event and for inviting me here today to talk about the menopause and the microbiome. So it's, as Katrina said, I'm the academic director for the Centre of Skin Sciences. That's a large academic centre within the University of Bradford, which is and we look at various aspects of skin biology, including immunology and inflammation, hair biology, wound healing, and aging. And I'm also the scientific director for the Plastic Surgery and Burns Research Unit, which was set up in the aftermath of the Bradford City Fire disaster. So I work with plastic surgeons, with clinicians, and with dermatologists, so we have access to human skin and tissue. And my career, I'm very particularly interested in aging, in skin aging, and in hair biology, but particularly from the view of the steroid hormones, the effect it has on the skin and the hair follicle. So the menopause is an example of programmed aging in the female. So it occurs about the age of 50 when the ovary stops producing estrogen. So estrogen will rise at puberty and fluctuate throughout the menstrual cycle, can be very high during pregnancy. And then about the age of 50, it stops being produced. And this is not a gradual reduction. This is a falling off the cliff a drop in estrogen. But prior to that, from about the age of 40, women could go through what's called the perimenopause, where they're still having their menstrual periods, but these changes are starting to occur. So women may experience symptoms that are associated with menopause, like hot flashes, mood swings, etc., that may last for anything up to 10 years. But more importantly is the effect it has on other tissues, because estrogen is a target hormone for more than the reproductive tissues. So it affects just like the cardiovascular system. So after menopause, the protective role of estrogen is lost in postmenopausal women. So they're at actually higher risk of cardiovascular disease than men. So it has effects on other tissues like the immune system, cognitive function, and of course, what we're very interested in is its effect on the skin. So with as we see with aging scalp, we see huge differences in the structure of the skin. But there are issues around particularly hormone replacement therapy because of its association with breast cancer. So in terms of the female life course and the microbiome, during the life of a woman, you see these very distinct hormonal states that drive the overall physiology of both the host and the microbes. And as we've already heard earlier, that we see these changes in these interactions that can drive some pathologies like bacterial vaginosis, but also they can drive other things like diabetes, polycystic ovary syndrome, and obesity. And particularly over the life course of women, from sort of puberty through reproductive age through the menopause, there's very large gaps in our current knowledge. And this was interesting because what I did was to do a search of menopause in the microbiome, and you can see that there's only sort of been a flurry of interest in this over probably the last 5 years or so. And then the number of articles that have been published under this, under menopause and microbiome, is only 262. And so In terms of the skin microbiome, there's obviously much more, because we see these changes throughout skin that in pathology like eczema and acne, chronic wounds, cellulitis we have here. So you see this infection in underlying the skin, but with aging and there are starting to be some more studies on aging skin and the microbiome, and also the effect that the microbiome has on the hair. So in terms of the skin microbiome, there's about 3,500 articles, but when we add menopause into that, so skin microbiome and menopause, then we come up with just 8. So it is a very under-researched area. And so I kind of then looked at estrogen and the microbiome, and clearly there is a lot more work in this area, but it doesn't really define what estrogen is, because through our lifetime we have different, our body makes different types of estrogen that are more predominant at different times of our lives. So the one that binds to the estrogen receptors, estradiol, is the most common during reproductive ages, or the highest circulating estrogen, whereas estrone is the estrogen that we see in menopause. But even so, there was only 555 articles on estrogen and the microbiome, and within that, only 9 that specifically looked at skin. And this is very surprising, because skin is a very important estrogen target tissue. It increases epidermal proliferation and the turnover of collagen, enhances vascularization, influences the hair cycle and the sebaceous gland. And we know that it improves the rate and quality of wound healing. And a lot of this data has been gathered by by studies of hormone replacement therapy that clearly will impact at reducing these effects of that are seen when estrogen is lost after menopause. So we know it's an estrogen target tissue, and this goes right from the epidermis to the dermis, through the hair follicle to the vasculature. It expresses the estrogen receptors, both ER alpha and ER beta. So we know there are 2 intracellular estrogen receptors. So we can see it quite clearly here in the epidermis, the dermis, the hair follicle, and cultured cells still retain their expression of the estrogen receptors in vitro. Now, the 2 estrogen receptors, ER alpha and ER beta, they're not, went backwards. They're not isomers, but they're two separate proteins. And what's interesting is that their DNA binding domain, which is the bit that allows them to activate gene transcription, they share very strong homology at 97%. But their hormone binding domain, which allows them to bind to the ligand, like estrogen, they only share 60% homology. They bind estradiol with a similar affinity, but they can bind different types of estrogens to different affinities. So again, there's quite a lot of interest in understanding which compounds may activate estrogen receptor alpha, which is the one that's the predominant one in reproductive tissues, like the endometrium and the breast, whereas ER beta is one that's found in non-classical tissues, or predominant in non-classical tissues, like the skin. These are transcription factors, so they will interact directly with the DNA to their estrogen response elements, and the type of a reaction that you see, or proteins that are transcribed in response to those, very much is tissue specific, and this will depend upon tissue specific activators or repressors of that gene. There's also been shown that there is a non-genomic estrogen receptor that's found at the plasma membrane, because genomic receptors like ER alpha and ER beta are more slow acting. These you see a very rapid response to these G protein coupled receptors. So as well as the skin, obviously the vaginal epithelium is a very important estrogen target tissue. And as it has already been mentioned before, we see differences in the vaginal microbiome depending on ethnicity. Between puberty and or through reproductive life, you see an increase in estrogen, which is also high during pregnancy. And the rise in estrogen at puberty will cause the maturation of the vaginal epithelium. And again, we've heard a lot about the mucosa and the lactobacillus that act on the glycogen as a substrate to produce lactic acid and reduce the pH. Just as a side issue, we've recently got some proof of concept funding from NBIC to develop a smart biolayered tampon that's electrospun with a polymer to try and treat bacterial vaginosis. And again, this diagram just shows you the changes that we see in the vaginal microbiome through the life course. So in the infant, the lactobacillus will be high, and then pre-puberty, it's not there, and then you see it again at puberty through reproductive life, and then you see this significant change at menopause. And the dysbiosis of again, there's a lot of interest in the bacterial in the vaginal microbiome, because the dysbiosis not only results in conditions like bacterial vaginosis, but also can influence conception, and studies with IVF have shown that this can have an influence, miscarriage, preterm birth, and also endometriosis and female cancers of the reproductive system, like the cervix and the endometrium, and even the ovaries. So at menopause, you see very similar changes as you do in the skin in the vaginal epithelium. So with a reduction in estrogen, there's a thinning of the vaginal epithelium. There's a reduction in the glycogen production. So there's less nutrients for the lactobacilli, and so the number of those bacteria reduce, and you get the change in the pH and the opportunity for overgrowth of other bacteria, which can quite often result in a UTI. As well as the thinning of the epithelium, you also see changes in the elasticity of the tissue that, again, similar to what we see in the skin. We see a reduction in the collagen and the elastic fibers. And then this can lead to this atrophy of the tissue can lead to traumatization, infection, and pain. And again, it's very clear, as we see with the skin, when you replace the estrogen, and again, this can be either local or systemic, that you see you restore the tissue to as it was pre-menopause. Another, instead of giving estrogen, another one is to give the what we call is an androgen. This is an adrenal androgen, dehydroepiandrosterone, or DHEA for short. And because the vagina has the ability to metabolize DHEA into estrogens, it can make estrogen from this adrenal androgen, which will activate the estrogen receptors, and again will help to restore its function. Because it doesn't have aromatase, aromatase is the enzyme that's required to convert androgens into estrogens. Because the endometrium doesn't have aromatase, then it doesn't lead to the risk of stimulation of the endometrium, which is always a worry, that you are stimulating the endometrium in the absence of progesterone. So humans are very different from other mammals because they've during evolution, humans and some primates have developed this ability to secrete high levels of DHA from the adrenal cortex, which can be converted into androgens and estrogens. So prior to menopause, obviously the ovary is the main source of estrogen, which is released into peripheral tissues, but the adrenal gland also secretes high levels of DHEA, which can be converted to testosterone, estradiol, and DHT, depending in peripheral tissues, depending upon the levels of aromatase or 5-alpha reductase. But after menopause, estrogen is still produced, but in peripheral tissues from the circulating DHEA. So, but the tissues will depend upon whether they have the enzymes that are able to metabolize DHEA into androgens and estrogens. So basically, each tissue becomes the master of its sex steroid environment at the menopause. So there is this big area of intracrinology where tissues make their own and cells make their own androgens and estrogens from circulating steroids. And many of this is coming after menopause from the adrenal cortex. So, but the DHEA also decreases with age. You don't have the big drop off that you do with estrogen, but you have this gradual decline. So when people are in their 70s or 80s, they have very little DHEA. So estradiol, or the ability to make the estradiol, is very much reduced. And again, looking at the role of DHEA in the microbiome, there's only 28 articles that are published, and most of these are around the area of polycystic ovary syndrome. If you include the skin or vaginal epithelium into that, then there are absolutely none. So then I thought perhaps we can learn a few lessons from the gut. And we know that the gut, the bacteria in the gut are capable of metabolizing and modulating circulating estrogen. And estrogen, when the liver conjugates the estrogens that are removed, but the gut is able to either increase free estrogen or decrease it depending upon the enzymes there, and this is called the estrobolome. And so here you see estrone, which is the major circulating estrogen at menopause. It can be converted to estradiol if there's aromatase present, but the microbiome in the gut has or some bacteria can express the beta glucuronidase, which will convert these conjugated estrogens back into their active form. So the gut microbiome can regulate the amount of estrogen present. And again, this may have impact if they're increasing the free estrogen on conditions like endometriosis. And there's even been some links to suggestion that it may be linked to breast cancer. So, and this is a very nice review that they show all the different influence that the gut microbiome axis can have on the on whether there's more estrogen there or there's less estrogen there. And again, this is the breast cancer has been linked to that because about 74% of breast cancers are estrogen receptor positive. And there's also been a very nice study showing the the sexual dimorphism of the gut microbiota and the influence of menopause. So this was measuring estrogen and estradiol. So the first one is premenopausal women, the second one is postmenopausal women, and the third one is men, so that you can see that menopause really in women really reflects the gut microbiome of men. So the skin, there's clearly a skin gut axis in the microbiome, but again, in terms of articles, there's only 75 that have been published. And again, once you add menopause into that mix and you add estrogen into that mix, there's nothing. And even when you add HRT into that search engine, there's still nothing. So it is a very underdeveloped and under-researched area. This just highlights the complexity of estrogen signaling, that it can be via these genomic receptors, or it can be by the membrane receptors, or it can even be receptor independent, because estrogen has a phenol ring. It can act as an antioxidant. And then we also now know we have this additional influence of the estrobolome of the microbiome. So I haven't really got a summary of, sorry, I don't know why it keeps doing that. I don't really have a summary of the role of menopause in the microbiome. I just really have a lot of questions. And the fact that that it isn't really understood, but also, you know, whether the role of the microbiome in regulating estrogen and what the impact of different HRT regimes on the microbiome may have, including DHEA, selective estrogen receptor modulators. I haven't really mentioned those. Those are agents that will activate estrogen action in one tissue, but so there'll be an agonist in one tissue and an antagonist in another. The classic example of that is tamoxifen, that's given for breast cancer treatment, because it will be an antagonist in breast but an agonist in bone. There's now this new area of research into native estrogens with selective action in tissues that are being looked at for their role in for the use in HRT and in contraceptives. And we also know that phytoestrogens can interact with the estrogen receptors. So all of these are still, but how they interact with the microbiome is unclear. And in terms of women that are being treated for breast cancer, tamoxifen I've already mentioned, but aromatase inhibitors are basically inducing the menopause because they will inhibit all production of estrogen. And then the impact of the gut-skin axis at menopause. Again, there seems to be very little understanding of what happens when we see this dramatic change and loss of estrogen at menopause. So thank you very much.